Hi, my name is Emily Yocom. I'm currently a biology major at Kentucky Wesleyan College, and I'm presenting to you today on my experience in the Cooper Lab at the University of Kentucky during the KBRIN summer program in 2015. Now, while we get a lot done in our lab research-wise, we also have the motto of work hard, play hard, because we believe you always need to have some fun. And pictured here is a photo from when we went on a lab hike to Natural Bridge. We're also involved in a fair amount of outreach programs because what are we if we're not helping those of the future learn? Just to generally go over some of the outreach programs we've been involved with, we've helped out with multiple STEM camps. One specifically coming up is just for young girls. We've also worked with 5th through 8th graders in a STEM Blue camp and at a health fair for Kentucky teachers. We've also worked at 2 different youth science summits, one in Lexington at Lexmark and another coming up in Louisville. Also, as I mentioned previously, we've taken 3 lab hiking trips to Raven Run, Natural Bridge, and Berea, and I've also had the ability to review papers for journals such as the International Journal of Zoological Research, Frontiers in Pharmacology, and the Journal of Neurophysiology. Pictured here are some photos from our first 3 outreach programs with the STEM Blue Camp, our Teacher Health Fair, and the Lexmark Youth Science Summit. These are photographs from our lab hike that we took to Raven Run. And finally, pictures from our hike to Natural Bridge. The photo to the left of your screen was actually taken on the top of Natural Bridge and is a hike I would highly recommend. Finally, before I talk about our specific research, I attended a fair amount of speakers and symposiums this summer because Dr. Cooper insists that there is always something more you can learn. This summer we've heard from a Nobel laureate, multiple PhDs, and postdoctoral students, as well as hearing 2 separate doctoral dissertations. This is where our specific research project begins. Since we are in a very research-dense lab, there are 4 major projects which I've been a part of this summer. I will speak to you on the first 2 that are highlighted in red, and my partner Jacob Cyphers will cover the last 2 in blue. The first project I'm going to speak to you on is the effects of cold exposure and influence of modulators on the larval Drosophila heart. It's the research of Yu-Chen Zhu, one of our graduate students, and the paper is currently being submitted for review. Just for a general overview, Drosophila are ectothermic organisms which are very susceptible to temperature changes in relation to their survival. However, the Drosophila has shown to be quite resilient to varying rates of cold shock. For example, Drosophila in nature are known to survive cold snaps that may occur. Due to this novelty of survival, the Drosophila has been a great model for genetic and metabolic experiments to try and pinpoint these exact genetic advantages or physiological changes that may occur. To the right of your screen, I have a picture of the life cycle of Drosophila. The main stage that we worked with was 3rd instar, which is pictured right here at the bottom of your screen. A fair amount of research has also been done on Drosophila in relation to cold exposure. For example, by Chen and Walker on a review made in 1994, they discovered that Drosophila have a limited survival rate in prolonged and extreme cold exposures. In fact, they held their larvae and adult flies at 0 degrees Celsius for up to 20 days, and very few, if any, of their flies actually survived. So while it may seem that the cold kills Drosophila, Drosophila are known to have a global presence in a multitude of environments. This is where the main question comes in as to why are they surviving? What's happening? What's going on? Well, in another paper by Kelty and Lee published in 1999, they discovered that a process of rapid cold hardening, or RCH, suspects that there are physiological changes or different cryoprotectants that are present in the flies or in their hemolymph that are preventing them from freezing in acute cold. Specific ones that were tested were HSP70 and carbohydrates, which were found to not increase during freezing, but according to Overgaard in his 2007 study, there is something out there that's reducing the rate of apoptosis. His leading idea was that increasing the osmolality of the hemolymph would decrease the rate of freezing. This being said, what we tended to focus on were different modulators that were present in that hemolymph that could change osmolality. In fact, little attention has been given to the actual physiological role of modulators present in the hemolymph. This is where our study comes in as a novel and new idea. The 4 modulators that we chose to study were serotonin, octopamine, dopamine, and acetylcholine, which are all known to rapidly alter physiological function. In fact, all 4 of these modulators are also known for regulating cardiac muscle function and being a stress response. This helps our study because we're studying specific heart rate changes in cold exposure. Therefore, cardiac muscle function has to deal with the heart rate, and stress response would be how they respond to acute or chronic cold shock. The most important thing in a study is proving that it's significant, and our study tends to be significant because it allows us to get a better understanding of the role of modulators in allowing Drosophila to survive seasonal changes in their natural environments. And discovering the role of these modulators may open investigations into the mechanisms of maintaining physiological functions in other invertebrates undergoing cold exposure. As you've heard me mention a couple times now, I've said the word cold a lot, which provides the fact that our methods tended to take place in a cold room. In fact, our exact methods were that we had 2 different stock supplies of Cantonese flies, one which was held at 21 degrees Celsius, and another which selected second instar Cantonese flies from the 21 degree stock and moved them into a 10-degree cold room so that they could mature for 10 days to third instars. Once both stock solutions had reached third instar stage, they were ready for our 2 separate tests that we did. The first test was to measure heart rates while moving from room temperature to cold. This being said, we would do a dissection of the third instar larva in the 21 degree Celsius room, and then take an initial heart rate when they were applied with fly saline, which was a special development by Clara de Castro, who is another member of our lab. After the initial heart rate at 21 degrees was taken, the tray was moved directly into the 10-degree cold room, where the saline was removed, and either another round of saline or a different modulator was tested. For the cold-to-cold exposures, all work was done in the 10 degrees Celsius cold room, including dissection, first application of saline, removal, and application of either the second saline round or the modulator. Pictured here is the actual dissection, which we did using a very fine pair of scissors and to cut through the cuticle, and a set of pins which separated the cuticle out so we could get a better view of the larval heart. This is the actual view you would get of a third instar Cantonese larva underneath the microscope, and it was dissected so that the heart would be visible and so that modulators could be directly applied to the heart. A separate section of whole animal measurements were also used via the ant farm technique by Henry Arrudo, a previous lab student. Finally, our main measurement was by taking the heart rate of the larva. The larval heart is located on the dorsal side of the body near the posterior spiracles, which, as you can see, are the 2 brown dots near the top of your screen. The heart tends to be situated between the 2 trachea, which are the clear tubes. And the larval heart is also myogenic, which means it beats on its own, and is bathed in hemolymph, allowing for the direct effect of different modulators to be measured. Now, if you think about it, hemolymph is like the blood of a larva. Since they have an open circulatory system, the hemolymph just washes over the heart, which is why I'm saying the direct effects of modulators could be measured when we replace the hemolymph with this modulator. Heartbeats were counted for 1 minute after being treated with saline, and then for 1 minute after being treated with the modulator in question. And as I mentioned, the heart is located right about here, and there's also an aortic tube right here that pushes the hemolymph back down towards the head. Due to us trying so many different things, we acquired a fair amount of data. The main graphs I'm going to talk about on this very busy page are the 3 that are highlighted in red on the left. However, I'm going to go over quickly the other graphs that are on this page. As you can see, across the board, almost all of the heart rates dropped when they were moved from room temperature to cold. The possible explanation of this could be that protein rigidity is slowing down the metabolic rate. Here we have the 3 most important graphs I could find from room temperature to cold movements. The first is a whole larva room temperature to cold count. As you can see, every single sample decreased its heart rate. The middle is an in situ version applying a saline exchange while moving from room temperature to cold, and once again, every single heart rate dropped. You may question why the saline exchange and the whole larva heartbeats are different, and there's a possibility that it could be just that the heart rate is easier to count on the in situ animal rather than the whole animal, which is possibly moving due to the ant farm technique. The final graph that I'm going to talk about is all the way on the right of your screen, and it's that of serotonin and moving from room temperature to cold. As you can see, serotonin showed our greatest amount of data in the fact that it had a majority lack of change, and was our only modulator that did not show a significant decrease when moving from room temperature to cold temperature. Now that we've seen all the room temperature to cold data, here's our cold to cold data. The first thing I'm going to mention as well is that the main graphs that we're going to go over once again are the 3 highlighted in red to the left of your screen. Looking at the other charts, however, the main trends are that a majority of the heart rates increase when treated with different modulators in cold to cold. The 2 graphs that you may question are the 10 micromolar octopamine and the 1 micromolar dopamine, which both slow the heart rates almost to 0 in both of their samples. We're not sure on the exact mechanisms, but it's a possibility that 10 micromolars octopamine was too much and was an abnormal presence that could for the animal and led for the heart to not be able to start. And the opposite could be true of dopamine, in that 1 micromolar was not enough to start all of their heart rates. In these main 3, I'm going to start all the way on the left in the cold to room temperature saline exchange. Here we have a great increase in heart rate. This is also the only chart that is scaled on the y-axis to 300 beats per minute. The only reason this is scaled differently is because the 300 scale would compress the other data too much for them to all be scaled to the same size. You might question why these go up so much more than regular saline counts, and it could be because the protein structure is less rigid in the heat and it's easier for conformational change in ion channels to work. But it is showing that with heat shock, the heart does continue to beat. The middle graph shows a cold-to-cold saline exchange. This is a novel graph because, as you saw in the first charts, the room temperature to cold saline dropped significantly. Yet here in cold to cold saline, there is not much of a change, where if it was holding true of all the other modulators, there would be a significant increase. This will be described later. And finally, the third graph is a diagnostic run for serotonin. Serotonin in the cold-to-cold exchange showed our greatest increase in heart rate over any other modulator. Next, we did a cocktail. Running a cocktail was a new experience that had not been previously studied, and we ran it purely for the fact that there could be multiple modulators working in the hemolymph at one time. So we had a cocktail of 100 microliters of each of the 10 micromolar stock solutions of the 4 modulators to determine if there was a possibility that multiple modulators were working at one time in the heart rate. These graphs held true mostly to those of the other modulators, and the fact that moving from room temperature to cold caused a decrease in heart rate, while moving from cold to cold caused an increase in heart rate. The cold-to-cold cocktail actually showed the greatest percent change overall in the cold-to-cold measurements. Here's what I consider the most significant graph in the fact that it allows for a better numerical analysis. On the left of your screen, the first thing I'm going to point out is the vertical hash bar in the middle, right here. And that would be that of serotonin. As you can see, serotonin had the only positive percent change out of all of the modulators. The next thing I want to show you is the saline bar, which is right here, the first one with the wavy lines. And if you consider saline and its error bar, it encompasses all of the other modulators which are either within the error bar or are more negative, such as octopamine or dopamine. This being said, the only modulator that showed significant change moving from room temperature to cold would therefore be serotonin, since everything else was within the range of the saline. The second graph, the first bar I want you to look at, is right here, and is a bar you can barely see. That would be that of saline. And saline had a 0.076% change, which was a novel idea since it changed so much moving from room temperature to cold that certain mechanisms are holding the animal stable even with a saline switch at cold to cold. Next thing I need you to look at is this bar here, which is that of dopamine. Dopamine produced the only negative percent change moving from cold to cold. You may question how this then impacts the cocktail, which shown right here has the most positive percent change. How would this be possible if there is a negative modulator in the works? We believe that the positive effects of serotonin in the other positive modulators are then outweighing the inhibitory effects of dopamine and allowing for the cocktail to have the greatest percent change. As I mentioned previously, here is serotonin, which presents the most positive percent change of any of the modulators. So in summary, acute cold exposure has shown to decrease heart rate in larval Drosophila, and although dopamine, octopamine, and acetylcholine have all shown substantial heart rate when they're held only at substantial heart rate increase when they're held only at room temperature, although they decrease so drastically when moving from room temperature to cold. Serotonin is the only modulator that showed a substantial increase in both acute and chronic cold exposures, and serotonin in the cocktail overcame the inhibitory effects of dopamine. Therefore, from our studies, serotonin appears to be the dominant modulator in reducing the effect of cold shock and in long-term cold conditioning. A few future experiments that are currently in the works are cold shock resilience in arthropods, most likely crayfish, with modulator treatment, the rate and/or contractility of a heartbeat after enduring acute cold shock, since we noticed in our studies that some heartbeats tended to be stronger than others in the cold, with the effect of different modulators, the composition and role of proteins in hemolymph in acute and chronic cold shock, Currently, we're waiting on results from an HPLC, which is a high-performance liquid chromatography results of the hemolymph, which is studying the main 4 modulators that we picked. And this future study could show different compositions in the hemolymph other than just that of the 4 modulators which we picked. Next, this is a direct lead into the mechanisms behind the maintained heart rate after treatment with serotonin and the combined cocktail and changing temperatures. And one final experiment could be other possible combinations of cocktails. We used only the 4 modulators which are most studied and most well known. Therefore, that opens the door for possible other cocktails that could be studied, which we'll have a better idea of once we get back the HPLC results. And as I mentioned earlier, we were able to finish the initial data collection for this experiment during this 10-week program. The paper is currently being written, revised, and hopefully submitted for review. My final experiment I'm going to talk to you on is the effect of perturbing the cholinergic neurons in developing Drosophila larvae, which is the project of Cole Malloy, another graduate student, and is a proof of concept experiment. This experiment utilizes the Gal4 UAS system, which allows for only cholinergic neurons to be targeted. Now, cholinergic neurons are those that deal with acetylcholine, and acetylcholine is the main neurotransmitter for sensory neurons projecting into the central nervous system in Drosophila. In the Gal4 UAS system, Gal4 is the driver gene, whereas UAS controls the expressions of specific target genes. The 4 lines which we used were UAS TRIP, UAS SHY, CHA Gal4, and D42 Gal4. In this experiment, UAS TRIP was a temperature-sensitive ion channel used to regulate ion channels at changing temperatures and heat. UAS SHY blocks neuron transmission. CHA Gal4 is selected to target neurons that make acetylcholine or specific cholinergic neurons. And D42 Gal4 is a motor neuron driver. This allows for a proof of concept that these certain channels are shut down at heat, which is proven when the larvae stop moving. Our results are shown here in these 3 crosses right here, which are our controls, seeing as how each cross was crossed by Canton S to rule out any possible confounding variables. The room temperature control, body wall contraction controls are shown with the white bar, whereas our experimental temperature of 36.7 degrees is shown by the black bar. As you can see in all of our controls, when the larvae were incubated for 5 minutes at 36.7 degrees Celsius, their body wall contractions increased, which could be the possibility that their metabolism is increasing with the heat, thus there are more body wall contractions. And our concept was proven as well in our 2 cross lines with the fact that the UAS strip channels, when stimulated and activated by the heat, then dumped all of their calcium, which led to a almost seizure-like result in the larva, and their body wall contractions ceased after they had been heated to 36.7 degrees Celsius. Therefore, we attained the proof of concept that we were hoping for. You may notice that the UAS shine line is missing from our results, and that's because we were unable to obtain virgin females for an initial cross. Here's a list of my references. And for a few acknowledgments at the end, I'd like to thank all the members of Dr. Cooper's lab, including Dr. Cooper himself. It's been a pleasure to work with him, and I couldn't have asked for a better mentor this summer, as well as all the graduate students for keeping me busy for these past 10 weeks and never giving me a break or time to rest. I'd also like to thank my lab partner, Jacob Cyphers, and the KBRIN program, which actually funded my research, as well as my college, Kentucky Wesleyan, and the University of Kentucky for having me here. If you have any questions, please feel free to contact me at EmilyYo@kwc.edu, EmilyYo@kwc.edu, or Dr. Cooper at the University of Kentucky if you have any questions about his lab. Thank you.